Hello all. We were discussing about the urine formation, and we have said that there are three steps in the urine formation. First one is the glomerular ultrafiltration of plasma, which occurs at renal corpuscle. Second is the tubular reabsorption of water and solutes, and third is the tubular secretion of selected solutes into the tubular fluid. So in the previous session, we have detailed the first step, glomerular ultrafiltration. Before proceeding to the tubular reabsorption and secretion, we'll look into the common principles and mechanisms that aids in these tubular functions. Once the filtrate is formed in the renal corpuscle, it will be passed down into the renal tubules, and there many qualitative as well as quantitative changes occur. That is, the solutes and water will be reabsorbed from the tubular fluid back into the tubular epithelial cells, or certain substances will be secreted into the tubular fluid. And these happens by means of various transport mechanisms. And we can see that these transport mechanisms are very selective. That is, they are specific to their solvent or solute. Each solute and water are having specific transport mechanisms of their own. Whereas in the glomerular filtration process, it was the same for both water and solutes. So we can say that glomerular filtration is a non-specific process because the mechanism of filtration is not different for water and the various solutes there. Whereas tubular transports are selective processes because they are having different transport mechanisms for different solutes and water, and it varies in the different regions of the tubule. And this tubular exchange of water and electrolytes finally helps in the determination of the volume and concentration of the urine. And this glomerular filtration rate is about 125 milliliters per minute or 180 liters per day, whereas the quantity of urine excreted out is about 1.5 liters. That is, it forms only less than 1% of the glomerular filtrate. So a large quantity of water and solutes are reabsorbed by the time this glomerular filtrate forms urine. So we'll look into it in detail. So when the glomerular filtrate reaches the renal tubules, there the water and other solutes will be reabsorbed by the tubular epithelial cells, and from these epithelial cells the reabsorbed substances will move into the interstitial fluid of renal medulla, and from there they move into the blood in the peritubular capillaries. Since these substances are taken back into the blood from the glomerular filtrate, the entire process is called tubular reabsorption. And we can see that usually glucose and bicarbonates are almost completely reabsorbed from the tubular filtrate, and water, sodium and chloride, about 99% are reabsorbed from this tubular fluid. More than 90% of the potassium and about 50% of the urea are reabsorbed from the tubular fluid. The transport mechanisms of different substances vary in different parts of the renal tubules. Broadly, they can be classified into passive and active transport. You know what a passive and active transport is. So passive transport is the movement of molecules along the electrochemical gradient or downhill movement without any expenditure of energy, whereas active transport is the movement of molecule against the electrochemical gradient, utilizing energy by the expenditure of ATP. The transport of solutes involves both passive and active processes, but water reabsorption is always a passive phenomenon. Okay. So we can see that chloride, urea, water, etc. are the substances that are reabsorbed passively, and sodium ion, calcium ion, potassium ion, glucose, amino acids, ketone bodies, uric acids, phosphates, sulfates, these all are transported actively. Routes of reabsorption. The reabsorption of substances from tubular lumen into the peritubular capillary occurs by two means. They are transcellular route and paracellular route. In transcellular route, the transport of substances occurs through the cell, whereas in paracellular route, the substances move through the space in between the cells. So in transcellular route, the substances are transported from the tubular lumen into the tubular cell through apical surface of the cell membrane. See, the tubular cell is having two surfaces, apical surface and basolateral surface. The surface that faces the lumen is called luminal surface or apical surface, and the surface opposite to it is basolateral surface. So from the tubular lumen, the substances enter the epithelial cell through this apical surface and it exits through the basolateral surface, and thus it enters the interstitial fluid. From the fluid, it enters the capillary. Transport of sodium, glucose, etc. occurs through this transcellular route. In tubular epithelial cells, nearer to the apical membrane, there are tight junctions in between the cells, and next to them lie the lateral intercellular space where the interstitial fluid is present. These tight junctions are actually not so tight, they have leaky channels. So when the transport of solutes and water occurs between the cells through the tight junctions and these lateral intercellular space, the process is called transport across paracellular pathway. Reabsorption of calcium and potassium ions from the proximal tubules. Likewise, reabsorption of water and some sodium ions are occurring through this paracellular pathway. Substances are reabsorbed from the tubular fluid in almost all the segments of the renal tubules, and proximal convoluted tubule contributes majority of reabsorption. Because of its structural peculiarity, we have seen that the epithelium of PCT is formed of brush-bordered cuboidal epithelial cells. So this brush-bordered epithelium increases the surface area, which facilitates the reabsorption, and this majority of the reabsorption occurs through PCT. Substances reabsorbed through PCT include glucose, amino acids, sodium ion, potassium ion, calcium ion, phosphates, chlorides, urea, uric acid, water, etc. And that are absorbed through the loop of Henle include sodium ion, chloride, potassium, magnesium, and calcium ions. And that through the distal convoluted tubule include sodium, chloride, and water. And through the collecting duct, it includes urea reabsorption. Then, if aldosterone is present, sodium will be reabsorbed, and if ADH is present, water will be reabsorbed. Next is threshold substances. That is, depending on the degree of reabsorption, substances are classified into three. They are high threshold substances, low threshold substances, and non-threshold substances. High threshold substances are those substances which are completely reabsorbed from the renal tubules and are normally absent in the urine, and they appear only if their plasma concentration becomes abnormally high, or if due to some renal diseases their reabsorption is affected. So they are called high threshold substances. For example, glucose, amino acids, ketone bodies, etc. They are completely reabsorbed from the tubular fluid and are absent in the normal urine. Low threshold substances are those substances which appear in the urine even in normal conditions. That is, they are absorbed only to a certain extent. For example, urea, uric acid, phosphate, etc. And these substances will be present in a normal urine. So they are called low threshold substances. Non-threshold substances are those substances which are excreted in urine whenever they are produced in the body. That is, irrespective of their plasma concentration, they will be eliminated and they are not at all reabsorbed. Metabolic end products like creatinine are non-threshold substances. Next comes an important point, transport maximum or TM. We have seen many substances are reabsorbed from the tubular fluid, and the rate at which maximum amount of a particular substance is reabsorbed from the renal tubule is called transport maximum. Or in other words, it is the rate at which the tubule maximally transports a particular solute. Beyond that limit, it cannot further increase the rate of transport of that substance, however high the concentration of that solute is. So whenever the concentration of the solute in the tubular fluid is more than the TM concentration, the transport mechanism becomes saturated and that particular substance will appear in the urine. So the effective reabsorption of that particular substance is affected and that will appear in the urine. So the TM value for glucose is 375 milligram per minute in males and 300 milligram per minute in females. Next comes tubular load. The quantity of a solute filtered by the glomerulocapsular filtering barrier and that is presented to the tubular fluid is called tubular load. And this tubular load determines the amount of substance to be reabsorbed from the tubule. See, the amount of the substance delivered to the tubular fluid in a unit time definitely will have a role in determining the maximum quantity of the substance to be reabsorbed. So it determines the transport maximum. And this transport maximum in turn depends on plasma concentration of the solute and the rate of glomerular filtration. See, the transport maximum value for glucose is 375 milligrams per minute and glomerular filtration rate is 125 milliliter per minute. So for glucose to have a transport maximum of 375 milligram per minute at a glomerular filtration rate of 125 milliliter per minute, the plasma concentration should be 300 milligram percentage. That is, beyond this 300 milligram percentage, glucose should appear in urine. But we know that when the glucose concentration in the plasma goes beyond 180 milligram percentage, this glucose will appear in urine usually. So this difference is called renal splay. See, renal splay is the difference between the transport maximum or the saturation and the renal threshold. Or it can be said that it is the concentration difference between the substance maximum renal absorption and appearance in urine. So renal splay is the difference between transport maximum and renal threshold. Now let's see what renal threshold is. Renal threshold is the plasma concentration at which a substance first appears in urine. See, for every substance there is a transport maximum value. Likewise, it also has a threshold level in the blood plasma. Below this threshold level, the substance will be completely reabsorbed and it will not appear in urine. But when the concentration of that substance goes beyond this threshold level, the excess amount of that substance will not be reabsorbed and hence it appears in urine. So this level is called the renal threshold of that particular substance. The renal threshold for glucose is 180 milligrams per deciliter. That is, the glucose will be completely reabsorbed from the tubular fluid if its concentration is below 180 milligrams per deciliter and hence glucose will be absent in the urine. But once the concentration goes above 180 milligrams per deciliter, the reabsorption will not be complete and glucose will appear in the urine. Thus, glycosuria occurs when the plasma concentration of glucose is above 180 milligram percentage. Okay. So, transport maximum is the maximal rate at which a particular substance is reabsorbed from the renal tubules, and renal threshold is the concentration of a particular solute in the plasma at or above which that solute first appears in urine. Okay. The regulation of tubular reabsorption is by three factors: glomerulotubular balance, hormonal factors, and nervous factors. Let's see them one by one. Glomerulotubular balance is the balance between glomerular filtration and the reabsorption of solutes and water from the tubules. When the filtration rate increases, the tubular load of solutes and water also increases, and this in turn increases the reabsorption from the tubules. And there are two possible mechanisms for glomerulotubular balance. First is the increase in osmotic and hydrostatic pressure in the peritubular capillaries, and second is because of the increased tubular load. See, when the glomerular filtration rate increases, more amount of plasma proteins will accumulate in the glomerulus. So this results in increase in osmotic pressure in the blood, and when it reaches the efferent arteriole, there will be higher osmotic pressure, and by the time it reaches the peritubular capillaries, it will further increase. So the elevated osmotic pressure in the peritubular capillaries increases reabsorption of sodium and water from the renal tubules into the blood. An increased load on the tubule. With the increased filtration rate, the load of sodium, glucose, and amino acid like substances on the tubule increases. More amount of sodium ions will be reabsorbed from the proximal convoluted tubule, and this sodium reabsorption is associated with increase in glucose and amino acid reabsorption because of co-transport. Thus, more solutes are reabsorbed and the balance is maintained. Glomerulotubular balance is the balance between glomerular filtration and the reabsorption of solutes and water from the renal tubules. When the filtration rate increases, the amount of solutes and water filtered will be increased. That is, the tubular load will be increased, and this increased load should be reabsorbed back in order to maintain the balance. Okay, so when the filtration rate increases, the tubular load increases, and this in turn increases the tubular reabsorption. So there are two possible mechanisms in order to maintain the glomerulotubular balance. First is because of the increased osmotic pressure in the peritubular capillaries, and the second is because of the increased tubular load. So let's see the increase in osmotic pressure in peritubular capillaries. When the glomerular filtration rate increases, the amount of plasma proteins that is accumulated in the glomerulus also increases. Okay, as the protein is not filtered, this protein will remain in the blood in the glomerulus, and thus the protein concentration will keep increasing as the filtration goes on. So consequently, the osmotic pressure increases in the blood by the time it reaches the efferent arteriole, and it will be higher in the peritubular capillaries. So this higher osmotic pressure in the peritubular capillaries increases the reabsorption of sodium and water from the tubules into the peritubular capillary blood. That is the first cause or first mechanism. Next is the increased tubular load. See, when the filtration rate increases, the tubular load, especially that of sodium, glucose, and amino acids on the tubule, will be increased. And this sodium reabsorption is always associated with reabsorption of glucose and amino acid through co-transport mechanism. So increased sodium reabsorption means increased glucose and amino acid reabsorption through the same carrier proteins. Thus, more solutes will be reabsorbed, and thus the balance will be maintained. Next is hormonal factors that regulate the tubular reabsorption. Here, the hormones which regulate the filtration rate and thereby the tubular reabsorption are listed. The aldosterone and angiotensin 2 increases the sodium reabsorption in different segments of the tubules. ADH increases water reabsorption from DCT and collecting duct. ANP and BNP decreases sodium reabsorption. Parathormone increases calcium, magnesium and hydrogen reabsorption, whereas it decreases the phosphate reabsorption. Calcitonin decreases calcium reabsorption. Third is the nervous factors that regulate the tubular reabsorption. So the activation of sympathetic nervous system increases tubular reabsorption. Also, indirectly it increases by stimulating renin secretion. We know that renin causes formation of angiotensin 2, and this angiotensin 2 increases the reabsorption of sodium from tubules. So the sympathetic stimulation causes increase in reabsorption of solutes from the tubules. These all are the common principles of tubular functions. Actually, this is just a teaser of the detailed tubular functions which we'll be dealing in the upcoming session. So, hope we'll be connected soon. Be safe. Thank you.